So hello, everybody, and thank you so much for joining us. Whether you're a PaleoPerks regular or this might be your first time joining us, we're really happy to have you here. Today, we're going to be hearing from Iban Goñi from the Université de Lille about outgrowths on Cambrian echinoderms, traces of the oldest parasitic interaction on deuterostomes. A little bit about how today's seminar will run. I'm beginning with these welcome and announcements, then we'll have a 30-minute talk. We have time for a moderated Q&A, so you can send in your questions at that point to our questions at PaleoPerks host through the direct message function, and that host this morning is Aharna. And then if you'd like to stick around to ask Ivan more about his work and his path to doing the research that he's doing today, you can stick around for a tea time and we can have more of a chat. Just a bit of housekeeping first. PaleoPerks values the participation of all folks interested in the paleosciences. You should have had to agree to the code of conduct in order to join this morning, or whatever time it may be for you. So please remember to abide by our code of conduct during today's seminar. We ask that you remain muted for the duration of the talk. If you have questions at any time, you can send those to the questions at PaleoPerks host, and we'll collect those for the Q&A at the end. Or when we're in the Q&A session, you can raise your hand. Also, if there's any technical issues that rise up during the talk, you can send those to the questions at PaleoPerks host. We have closed captions built into Zoom. You can use the CC button to show or hide them. We're going to drop a link in the chat in a moment. We'd love for you to nominate an outstanding early career researcher whose research you'd like to hear about during PaleoPerks. And we're also going to share with you a weekly feedback form for demographic information. This is anonymous, optional, and encouraged, so we'll have that in the chat momentarily. So today I'm really excited to introduce you all to Iban Goñi. Iban did his bachelor's in geosciences at Claude Bernard Lyon in France, his master's in géosciences at the École Normale Supérieure de Lyon, and is now a PhD student in earth science at the Université de Lille. So with that, I'll hand it over to Iban, who hopefully will forgive me for my poor French pronunciations.
Thank you for the introduction. Actually, the accent was pretty good. So I'll start sharing. Does it work? Perfect. And if I change the slide?
Great.
Oh, good. Perfect. So yeah, thank you for the introduction, and thank you for all of you for being here. And so yeah, as said, so now I'm a second year PhD student in Lille, so in another part of France. But actually, my PhD is not related to the research I'm about to talk with you. And so just for a brief context, so in spring 2022, I'll just put the pointer. Yep. So in spring 2022, I did my master project, but after that, I also did one additional year during which I did two internships, like two projects. And so one of which is actually the research I will talk about for the next 30 minutes. And so as some of you may recognize or not them, all of these are Cambrian fossils. So my main period, my favorite period of time is the Cambrian, and more especially actually the early and middle Cambrian. And so now I will only talk about these echinoderm plate fossils from the middle Cambrian. So all the information I will talk about are already actually published in a paper that was published a few months ago. So you could have a look later, but during this presentation, basically would be the same, but with more figures, more images. So it would be also much more fun. So basically, there are three main words in this title that we will consider for the rest of the presentation. So actually 3.5, because the first one is Cambrian, just for the period of time. The second one is echinoderm, but plus deuterostomes, so that's why 3.5 words. And the third one is parasitism. So Cambrian, most of you, or I hope all of you know this period of time. This is one of the oldest period where you can actually find fossils of animals. The first ones are certainly in the Ediacaran, but actually the Cambrian is also very famous because actually we find all of ancestors of modern animals, basically. And this period is actually essentially famous for these fossil Lagerstadt, actually like as the famous Burgess Shale in Canada, but also the Chengyang fauna actually that occurs in China. And in these faunas, so as I said, here is a quick representation of a phylogenetic tree for all animals. As you can see, all the stars, like the red stars, are modern animals that find their ancestors in the Cambrian. And actually, most of them are in the early or in the middle Cambrian. And among all of these stars, one of them represents deuterostomes. And deuterostomes, so as a quick reminder, deuterostomes are animals for which the first hole that actually appears during the development is the anus and not the mouth. Where the mouth appears first is for protostomes. And among deuterostomes, in modern ecosystems, we find echinoderms, hemichordates, lancelets, or cephalochordates, tunicates, and vertebrates ourselves. And here we'll only focus on echinoderms, but whether echinoderms or vertebrates, which are the most common of the deuterostomes, they find their first fossils during the Cambrian, and actually both of them are more or less around 520 or 21 million years ago. Nowadays, echinoderms are represented by 5 classes. So you have ophiuroids, sea urchins, sea cucumbers, starfish, and crinoids. But actually, in the fossil record, there were 25 classes in total. So 20 of them went extinct through time. And actually, most of them already appeared during the Cambrian. And so here, as you can see, so stage 3, stage 4, this is for the Cambrian. And that's approximately 515 to 510 million years ago. Also, what you can see is that during the Cambrian, there is not yet the pentaradiate symmetry that characterizes actually like modern echinoderms, like all the classes. But in the Cambrian, it was not the case. And actually, in the fossil record, we might recognize echinoderms as with the pentaradiate symmetry, but actually what characterizes echinoderms really in the fossil record is their skeleton. And so like little trigger warning for people who may have a trypophobia. But so now we change the slide. What actually characterizes the echinoderms is their skeleton, which is called the stereum, and which is a 3-dimensional mesh-like structure, basically. So you have holes and the skeleton. And so just here with a quick image, but one of the material that we have in our study is this plate, and there is no doubt that this is an echinoderm plate due to the presence of the stereum. And then the last word, so we did the Cambrian, we did echinoderms and deuterostomes, and the last one is parasitism. Parasitism is one of the 6 symbiotic interaction that exists among between animals or plants or whatever, among life actually. And all of the interaction are characterized by the positive, neutral, or negative effect they have on both symbionts, on both organisms. So when actually the interaction is positive to both of them, this is called mutualism, and this actually represents most of the pollinator and flower interaction. But this is also the case here for this fish and this sea anemone. The fish is protected by the predators, from the predators by the sea anemone, and also the fish repulses the other fishes that may want to eat the sea anemone by being just around it. Commensalism is another interaction where that's positive for one of the organisms but neutral for the other one. Here you have an example of a bird called cactus wren. So this bird actually builds its nest on the top of a cactus so that actually predators cannot reach the nest and the eggs actually. But for the cactus, the presence of the nest is supposed to be neutral, so there is no specific effect. When it's positive for one but negative for the other one, here you have, for example, parasitism. So here's a modern example of a fungus actually parasiting an ant, so that actually at the end of the life of the fungus, it can reach the top of a plant and disseminate its spores. In another case of positive negative interaction can be also predation, but actually in predation it's positive for the predator, but for a very short period of time. After it kills its prey, then that's the end for the prey. Whereas for a parasitic interaction, most of the time, the parasite needs the host to be alive as long as possible, as the parasite depends on its host to live itself. Then when it's neutral to both animals, both organisms, there's not much more to say about that. They're just like here, and it's fine for everyone. It's like the most peaceful interaction that exists. Then when that's negative for one and neutral for the other one, it's called amensalism. And here is an example of a tree whose roots actually create chemicals and that prevents other plants to actually grow. But there is no specific advantage for this tree actually to produce this chemical. So just like negative for other plants and neutral for this one. And then negative, it's the can be considered as the competition. So here I showed the competition between two organisms of the same species, but it can also be competition between species for like a same resource, for the same food, for example, and that also works. And so as you may notice, most of the interactions that are shown here on the screen, it would be really tricky to find them in the fossil record. Maybe you might be lucky and find the ant with the fungus directly itself in a number, but that's really rare. And also, for example, in the competition, the interaction can leave traces on the skin or on the surface of the animal, and you could also interpret the interaction through the traces that are left. And to me, there are three main ways to maybe reconstruct past interaction. It can be like just through inferences. You have herbivores, you have carnivores, and you can imagine and pretend the relation, the interaction they had, or through other modelization, for example. But most of the time, what would be like the easiest way to detect and see an interaction is to see it directly, actually. And here, for example, you have directly the interaction between a velociraptor and the protoceratops that was found in Mongolia. They were preserved that way. They were fossilized in that position. So we can see the interaction that directly. Or you can interpret the interaction through the traces that are left on the skeleton. And here is an example of a crinoid stem with a gall or a callus that was left by another organism through an interaction. And we are typically in this last situation with our echinoderm plates that I will talk about now. So all the outgrowths that you see here on the external surface of all of these echinoderm plates are traces that we interpret as being induced by an interaction. And so if you're not familiar with the echinoderm plates in the Cambrian, these are disarticulated plates. We do not have the entire organism. This is just a fragment of it. So it can be basically comparable to the bone of a vertebrate. It's not the entire vertebrate, it's only a fragment of it. So all the material that we studied here that I'm about to present, all the fragments, they come from one single bed from the Middle Cambrian here, so from the Woollyuan stage, actually. And so they come from one single bed, so the age is not completely precise, actually. And they all come from Australia, and more specifically, so in Mount Murray, which is actually at the eastern part of the sedimentary Georgiana Basin. And here, so basically, it's in Queensland. And so for those who are not familiar with microfossils from the Cambrian, there are many ways to actually look at Cambrian fossils. If you're lucky, you work with Lagerstadt fossils. But for other type of fossils that are super interesting too, of course, most of the time you are looking for marine carbonate rocks because actually life, like animal life, is exclusively marine at the time. And most of the time, so we are looking for carbonate rocks. And the thing is that in the early and middle Cambrian, essentially, there is a sort of taphonomic window where fossils were replaced by phosphate. And so Basically, our methodology is just to put the carbonate rock within acetic acid. So basically that's vinegar. And so you have the rock, you have the vinegar, and the vinegar, so the acetic acid dissolves the rock, but not the fossils because they are present with phosphate, and that does not dissolve with acetic acid. And so once fossils are separated from the rock, then since they are microfossils, so most of them are between like 100 micrometers to 2 millimeters long, you look at them under the microscope, and then we go through the picking process basically to put them on a stub, and then you put these fossils under the SEM, so scanning electron microscope, to get as much detail as possible. Of course, you want to see more details that are not possible to have through a basic microscope. And for this project, for this work, we also decided to put some of the plates through a CT scan actually, to look at them under the CT scan, because so we wanted to look also at the microstructure of these plates, to look at the stereome, to see what it looks like, and to see what kind of information we can basically have. And so 3 plates were scanned at the European Synchrotron Radiation Facility, so in France, and 4 of them were scanned in Ghent University in Belgium. And so we also did quantitative analysis thanks to Kenneth Debet, who is a researcher now in Warsaw in Poland, and who is like the specialist of paleoparasitology, basically, and who gave us advices of what kind of quantitative analysis to do on this material to get the information we wanted. And so we'll discuss about that later. But first, like, we wanted to look at the morphology of this structure and understand what they were. And the first result, let's say, is that so these outgrowths, we are looking at this and here as well. This type of structures, they are only on the external surface of the plate, only on the external surface, and this is completely unique for all Cambrian echinoderms that are currently known. So this cylindrical to domal shape with distal opening, it has never been found anywhere else during the Cambrian. So this is basically on the right, the typical type of echinoderm plates that you find in all microfossils from the Cambrian. When you have an echinoderm, this is what it looks like. There is no outgrowth. It's always smooth, let's say. But we know that in echinoderms, this kind of similar structures, they exist, but in other organisms, and they are actually a little bit different. The oldest, let's say, similar structure that we can find are these ones. So in C and G here, you have 2 types of sea urchins from the Ordovician. They are slightly different, but like rounded in shape. They have tubercles on the surface that were interpreted as sea urchins, and this one is actually pretty obvious. This one it's quite weird, but we're in the Ordovician, so anything's possible. And the thing is that, so in both cases, we have tubercles on the surface on each plate. So for example, this is one disarticulated plate, and you have the entire articulated animal. On one plate, for this one, for the upper one, you have tubercles, so outgrowth, that are all the same size, more or less, that are most of them exclusively in the middle of the plate, more or less, and there are either like 3, 4, or 5 outgrowths on the surface, but that's a regular pattern. You have the same shape, they are more or less all in the same position of the plate, and approximately the same number per plate as well. For the lower one is even more obvious. You only have like 2 tubercles on the surface. They have exactly the same shape, the same dimensions. They are exactly on the same position within one single plate. So that's super regular. And just for also more detail, actually, in modern sea urchins, we also know what these tubercles are. These are like, they represent attachment surface for the spine, actually, of each sea urchin. And also in the microstructure of these tubercles, there is also a regular pattern. Every time there's the external surface that is almost not perforate, there's almost no hole actually in the upper part. Then it's micro perforate, so tiny, tiny sterium with the tiny holes. And then the rest of the plate is also slightly different, but the sterium within a tubercle in echinoids, so in sea urchins, it's super regular within one organism, but also among one species. You can almost differentiate one species from another thanks to the microstructure. And there's also another type of tubercle I wanted to show you that's from a crinoid, a Devonian crinoid. And here you can also find sort of similar tubercles, but which are probably not supports for spines. We don't know, but it was just found like that. And once again, the dimensions are all the same. The position and distribution on the surface is actually quite irregular. The space between different tubercles is more or less the same. So there's something regular. There's a pattern that makes them not random actually. And so in our material, so in the plates from the Cambrian, you can see that actually it's quite different. So one thing is that some of the plates they have only one tubercle on the surface, one outgrowth, sorry. So here you have one, only one. So most of these ones are only have one outgrowth. But for some other plates, so this one has two, this one has two as well, this one has three, four, and even up to five. So it's not regular, at least, even though most of them only have one, it can be much more. The position is not regular either on the surface. The distribution of the outgrowth is not systematically the same from one plate to another. Also, the dimensions are not exactly the same. And so here you have the image of the plates, but here you have also the statistics, basically the numbers that come with these results. So basically, yeah, most of the plates that we have and that bear were on more tubercles. So most of them only have one. Some of them have two, but it can be up to five. Dimensions. So here is the frequency, the number of outgrowth, and here you have the diameter of the outgrowth. And you can see that the dimension is not the same from one outgrowth to another. It can be like only 80 micrometers up to 200. And we have exactly the same observation for the height. So there's a lot of variation. And also what you can see is that, for example, if we take an outgrowth, so a plate, sorry, that has four outgrowth. So it's actually here the dark blue. Some of them, so within one single plate, you also have outgrowth that have not the same height, so not the same dimension. So even within one single plate, we do not have the same dimensions. And here, so on the graphs on the right, what we wanted to represent was the distance from an outgrowth to the center of the plate, or the distance from the outgrowth from the edge of the plate, so the plate's border. And so what we observe here basically is that whether you have like 1, 2, 3, 4, 5 outgrowth on one single plate, all of them they are arranged in the same distances actually. The only thing is that for when you have only one outgrowth, most of the time they are close to the center, but they can be really actually far, even though we are like 400 micrometers. So actually, there is no specific position for the outgrowth on the plate. So they can be everywhere. There is no consistency in these results. So this is actually different from what we know in regular sea urchins that are the closest known echinoderms who have similar structure on the surface. And it seems to be completely different. So we have something different. And so we also wanted to look at the microstructure. There is actually quite less data in the fossil record for microstructure, but it's getting more and more done nowadays. And here what we observe as well is that so in modern sea urchins, the microstructure of tubercles that are support for the spines are strictly the same from within one single organism and even from one species to like within the single species as well. And here was what we can see is that from one plate to another that we scanned with outgrowth in the Cambrian, can be actually quite different. Here you can see that the microstructure is actually different within the outgrowth, but also a little bit below. So actually also there is a little bit of disruption actually within the plate. So the rest of the plate is actually also affected. But sometimes it's not at all. It seems like in this one, for example, the microstructure is actually the same within the outgrowth and within the plate. Here, for example, it seems like it's only in the outgrowth but not below that there is a different microstructure. So basically, there is no consistency from one plate to another, and we can actually we can see various things. In this one, for example, it's not that great because here we have a problem of preservation is not that well preserved, and here we lack the information. But once again, we lack this consistency in the microstructure. And so with another more recent example, uh, some similar outgrowth actually exists in modern echinoderms that are not actually supports for spines, so that are not tubercles known in sea urchins. But once again, here you can see that the microstructure is actually exactly the same, just from one outgrowth to another. It's just like a little bit denser, like so tiny holes and tiny stereome, but the limit within like between the outgrowth and the plates is strictly at the surface here, which is actually different from what we have. So it seems like we have something completely new. So with all the information we have so far and that we know, we consider that these outgrowths are nothing comparable to what we know that are generated normally, let's say, by echinoderms in the fossil record and even nowadays. So this is why we also came up with the symbiotic interaction interpretation. And then, so with a reminder of what the parasitism is, so that's a long-term relationship between two organisms in which an organism, the parasite, has benefits from the other organism, the host, harming it. But how do we show that actually this was a long-term relationship and not a short-term one? And how do we show the benefits and detriments of this interaction on both symbionts? So one thing for past actually parasitic interaction that has nothing to do yet with those three words, so long-term benefits and harm, is just that most of the time in other cases of parasitism occurrences is that they have a low prevalence in within the assemblage. So for example, you have a cranoid and mollusk interaction in the Jurassic, and you have like less than 10% of the cranoids that are parasitized by the mollusks. But most of them in the assemblage are actually pretty normal. And so that's usually, in most of cases of parasitic interactions in the past, this is the kind of things that we observe. And here actually our results also show the same. So in total, we have 120 plates, disarticulated plates with one or more outgrowth. And in total, so it represents less than 2% of all echinoderm fragments that we have in the assemblage, in the Australian assemblage. But to be a little bit more relevant, we only considered plates that were from the same area of the animal. So here I took thecal plates because all the plates that have outgrowths are thecal plates and they correspond to only one part of the animal. So for example, in our assemblage, we have also many stem fragments. We have many feeding appendage or just appendage fragments, but none of them, they show outgrowths. So we only considered similar plates, and even considering only these similar plates, we have less than 10% of the plates that have outgrowths. So we have more than 200 plates, 2,000 plates, sorry, that are completely normal. And so how do we show that? So coming back to the definition of parasitism, how do we show that this is a long-term relationship? So that's actually the only thing we have to say that is that if it were like a short-term interaction, like a prey-predator, usually the predator kills the prey. And so in that case, there is just no reaction from the prey. It just, it's dead, and then there is no reaction, no skeletal reaction, because actually nothing works anymore. But for a parasitic interaction, most of the time, there is a reaction of the host, and actually this is what we have here. we have outgrowths that are generated by the host, and so it was alive when it happened. And the fact that some of them can actually be really high, it means that at least the presence of the parasite actually was not um, harmful enough to kill the host. And sometimes, so since we have like 5 outgrowth on a single plate, it was not deadly, at least to the host. But the fact that there are different types of height, it means that at least it was a long-term relationship, but how long, I have no idea. So these are the kind of things that we should also better understand. And the problem is, so how do we know also if it's parasitic or not? And do we have like any modern relatives? Well, that's the problem because for any known, at least parasitic interaction involving an echinoderm, there is no outgrowth actually. So this type of reaction in modern echinoderms, it's not known at all. So we cannot compare with modern echinoderms. But the thing is that at least in the fossil record, there is like one structure that is that has been found. That's only in the fossil record, and that is called bioclustration. So bioclustration is basically the embedment of a symbiont, and so it consists in there's an animal, an organism that's the host, and another organism will come to the host, and but since like the host actually seems to be annoyed by the presence of the symbiont, he will try to embed it, so recover it to try to kill it. But so as you can see, there's always a distal opening at the end, so it seems like it can't actually recover entirely, but at least it tries to pushes the symbiont, the potential parasite, from the surface to yeah far from the surface. And the thing is that so this type of structure, it has been observed on brachiopods from the Cambrian, but also on bryozoans, on corals, on another brachipod from different periods. Here is the closest relative I found in modern environments, which is not exactly the same, but at least there is something a similar reaction. And the thing is that so we have that in the fossil record, but not on echinoderms. But it seems like it has been observed in other groups. But the real impact the interaction had on the host was not completely defined. Some researchers interpreted that as commensalism, some of them as parasitism, but it's hard to say. And in our material, the good thing is that at least we have 120 plates, and so it allows us to make just this simple analysis, just the size analysis. And what we observed is that so we also pictured and take measurements of normal plates from our same assemblage that have the same morphology and same microstructure, more or less, that we can see not from the CT scan, but just from the microscope. And the thing is that so all the plates that have outgrowth are generally much smaller than the plates that have no outgrowth. So we did it. We did this analysis, but we have to be very careful because the thing is that in our assemblage we are not even sure what class the fragments actually belong to. So the thing is that we are not even sure if we are comparing maybe just different species or just different organisms. Maybe the large ones are not comparable with the small ones, but at least we had to make this analysis to show that at least only the small, let's say, plates were bearing outgrowth, and that's a potential actually impact on the host, like on the size, on the growth. But how did it happen? We are not completely sure. And the problem is that the effect on the parasite, we have no idea. It seems like if it's actually due to in an interaction, uh, and if it was there, if the parasite was there, so at least and it was living, so at least it represented potentially an attachment surface, or it was actually able to live, to feed itself. So that's the positive impact, but we're not completely sure about that. That's our interpretation, and that was making more sense. But to know exactly, like, yeah, the attachment surface hypothesis also comes with another result, is that the shape of the plate that have an outgrowth, it's not different from the ones that have no outgrowth. So actually, there is no selection of the shape. Also, as we saw earlier, there is no selection of the position also of the parasite on the surface of the plate. So it's just like it seems to be it arrives and just lands wherever and starts to grow on the echinoderm directly. And so, yeah, in general, this is what we have. And so as yeah conclusions, so the main thing is that this type of structures is completely new for the phanerozoic record of echinoderms. So this was the most important things to say and to mention, at least to show that this actually exists. And so as detailed, the parasitism is probably, at least to us, the interpretation that makes more sense to understand actually how this outgrowth appeared and were generated. And if that's actually the case, that would represent the oldest parasitism in deuterostomes and in deuterostome animals. And so we're still waiting for this kind of interaction in chordates. And so as perspectives, there's still a need to identify the parasite and also the effect it had on the parasite. And so also in general, a better understanding of such structure, like how it can be generated. According to our results, it cannot be just generated like that by the echinoderm and required an interaction, but maybe something weird up in the Cambrian, and we never know. But and also what's for sure is that a greater attention to Cambrian articulated specimens would be great. And if we can find something similar in the fossil record within an articulated specimen, that would be great. And so just a quick parenthesis of what I actually do right now, since this is not a PhD research. Now I'm working on reef environments and reef ecosystems from the Cambrian, but still in the Cambrian, so that's still the period I'm interested in. So that you know, I'm still working in Lille here with Sébastien Clausen and Timothy Topper. And the last word actually goes to all the co-authors that actually made this project possible. Because honestly, this wouldn't have happened without them. Like they have many great advices that made it actually research possible and interesting. So my last word actually goes to them. And thank you for your listening.
Fantastic. That's so interesting. Thank you so much, Iban. Um, I think what you said about maybe weird things happened in the Cambrian, truer words were never said.
Mhm.
We'll now turn it over to the audience if folks have any questions. I see that we have a couple coming in already, so I'll copy and paste those, put those in the chat. So first, we have a question from Kartik, who's asking, have echinoderm plates apart from the outgrowth in the center changed much in morphology and ultrastructure in response to changing body symmetries?
Is it possible to repeat the question, please?
Yeah, it's in the chat as well. So they're asking, have
Okay, so
plates changed much in morphology and ultrastructure in response to changing body symmetries?
so in the ultrastructure, I'm not sure. Like because so for example in Cambrian echinoderms, so this kind of plates, actually the morphology and the symmetry, so the symmetry is not always obvious and not necessary, but this kind of thecal plates, the morphology seems to appear according to the previous plates that were actually already present. So these were apparently there were additional plates through the growth of the organism itself, and so this kind of plates were generated, and the shape depends on what are the other plates that are actually already exist, because all these okay, let me put a pointer back. All these notches here apparently were useful to let the organs to have access to the external part of the body. And actually, so there were soft tissues going through these notches, but actually you have to imagine like that this one here actually is going by pair with this one, and here that will make a sort of hole. And so let's say that this one appears first and this one in second. So the hole here would depend on the position of the previous ones. So that's a little bit of the morphology of plates in echinoderms during this time. And but for the ultrastructure, nowadays I don't know, but in the Cambrian actually there is not that many papers mentioning the microstructure of echinoderms. So there's not so much to compare with, but and also maybe that may also answer to the question. I think that indeed within the outgrowth, the ultrastructure will change for sure, depending on actually the size of the structure. So basically, here the outgrowth is much smaller and narrower than the rest of the plate, and so in that case, of course, the microstructure, the sterium is denser, just because the outgrowth is actually smaller. So it also respects that. And here also as well, for example, it's bended in this kind of notches. And here as well, the microstructure is also smaller because so it's folded, and so the microstructure also changes according to the morphology of the plate. So that's something that is observed. Yes. I don't know if that answers to the questions entirely, but I'm
Great,
still
thank you. And they can stick around for a tea time if they want to
yeah.
chat about that more. We have another question in the chat, which is, has this
Mhm.
kind of outgrowth been assigned to an ichnogenus or ichnospecies?
Can you repeat please again?
Yeah, so they're wanting to know if this kind of outgrowth that you've observed has been assigned to an ichnogenus or species.
Yeah, okay. Nope. No, we haven't named that. I know that it happens sometimes indeed as an ichnospecies, but no, we didn't. We didn't name that, and so we thought it was more like better to just let it like that and focus on the interpretation of it. But no, we did not name that structure.
And we have another question coming in here, which is, what is the relationship between the outgrowth size slash frequency on plates belonging to different echinoderms? So if there's a difference statistically between different hosts, can we conclude that encrusting organism is somewhat host specific?
Okay, so I'll read the question again. I'm not sure I understand.
If the person who asked this wants to unmute, they can feel free. My interpretation of this question is that they're asking, are there differences between which outgrowths you're finding on which kinds of echinoderm plates? So is there some kind of species specific thing happening there?
Yeah, okay. Okay, I see. Maybe, maybe I have no idea. The thing is that so that's really tricky to say because so I briefly say that I did not develop enough, but we do not even know what class all these plates belong to. We know that we know which classes are present within the assemblage. We have Edriosaurids, we have Blastozoans, we have another class as well. So we know we have them because we have specific plates of these different classes that are present in the assemblage. But the problem is that none of the specific plates that we have and that will help us identify the echinoderm have outgrowths. And the thing is that so these type of plates of thecal plates, they can be observed in each classes actually. Like this kind of plates are observed in all Cambrian echinoderms. This type of plates is not specific to even one class. So to try to identify to one species, actually, at the moment, it's not completely it's not possible. That would need very deep investigation, but this is not what we did here. But that could be super interesting to know. Definitely, that would be super interesting to know if that's like if the ones, for example, that have only three outgrowths are only what species. If the one that have like an outgrowth in the middle is another one. So I don't know, but yeah, that would be great to know.
Great. Yeah, that's interesting.
Mhm.
Um, Maybe a related question we have here is whether there is some sort of recruitment process involved. So you said that some of these plates have multiple outgrowths, and so this
Uh-huh.
person is asking whether if a whether plates with outgrowths are likely to have other plates with outgrowths near them, or are they all? separate. Are you seeing outgrowths that are next to each other on plates, or all the plates coming to you in a different form?
It really depends. It really depends. The only thing I might have noticed, and that was actually thinking that maybe this is not that random, is that, so for example, you have the one in Q here and the one in X, and so plates have more or less the same shape, and here you have two outgrowth there, and here they are more or less in the same position. So is it like completely random? I don't know. And is it specific to that shape? I don't know. Like, is this kind of shape is more likely to have one or only like two or four outgrowth instead of one or three? I don't know. Also, you can see that this kind of shapes that look like a like a Game Boy or something like a PlayStation board pad. They have like three outgrowth and have like more or less the same shape. But in these ones, the outgrowth are aligned, and in this one, it's like slightly curved. But why these ones have three and not four, or not only two? But actually, this one has two and not three. So there is no pattern, and that I found so far. So yeah, I don't know. I don't know. One thing is that indeed, when there are more than one outgrowth, usually they are never like super close to each other. There's always like a, let's say, minimum distance between them. Like there's never like one here and one just right next to here. So that's also a thing. But yeah, yeah.
Fascinating. Here's, I think,
Hmm.
this will be our last question coming up now. Maybe a related question kind of about the biology of these organisms, which is what are these parasites? So what methods could you use to identify the parasites, or is that even possible?
Very good question. Very good question. I know that that in so in other papers that found similar outgrowth, so on brachiopods. So okay, I'll go back to yeah, these ones. So usually in the papers they don't know. Like authors, they don't know what were the potential parasites or symbionts that generated these kind of structures. They might imagine tubular fossils that maybe were living within the hole, like within the hole that is generated, and maybe something that might have been I stay like with might have been another. I'm really not sure about that, but maybe like suspension feeding, and maybe stealing the food from the host, but that's very theoretical. And yeah, an organism that would just live there, stay there, and just yeah, feed on of whatever is passing by there. And something like tubular maybe, so as this closest modern, like more or less modern relative, there would be like helminths, so the kind of worms actually that were living in there. But that's, yeah, that's a very good question. And honestly, I found nothing in the literature that generated this kind of structure. But the problem is that we do not have modern relatives. So that's one of the main problem as well, to understand actually the ecology and the parasite actually. But for methods, good question. That would be something to try to understand. But which method use? Mm-hmm. Yeah, I don't know. But yeah.
This is forever the question of trace fossils.
Yes. Yeah, yeah.
Excellent. I'm going to take over the screen share again and just
Okay.
thank you again so much for that talk and for your really great answers. Next is going to be tea time with Iban. So our chat today, we can start off with a conversation about your Cambrian taxonomy hot take. So do you have any bold statements to say about who you think is related to who in the Cambrian? But first, we just want to thank you again for joining us. We're going to drop the weekly feedback form in the chat so that you can tell us about who you are and what you're looking for out of Paleo Perks. And we do want to invite you to our next talk, which is a week from today, and that's going to be a really special one. This is our 5th anniversary special seminar, so we're going to have all the co-founders of Paleo Perks there today: Elizabeth, Andy, Natasha, Chrissy, Jana, Kate, Pedro, and Rahemet. So we're really excited for that, and we hope that you'll join us. Now we're going to transition to tea time, but first we're going to take a little bio break. So we'll have a 2-minute timer. If you want to get up, take your camera off for a moment, and we'll come back for tea time.